Okay, now we're going to talk about organization of microbial genomes and then mechanisms of replication of DNA. Okay, so bacteria almost always have a single circular chromosome. A few species have been identified that have more than one chromosome, but it's very rare. And a few species have been identified that have linear chromosomes, but again, that's very rare as well. So the general organization is a single circular chromosome. Size range going from about 500,000 bases to up to 8 to 10 million bases, but that's sort of the extreme upper end. Typical sizes would be more like 2 to 5 million base pairs. 1,000,000 base pairs is called a megabase pair. Okay, 1,000 base pairs is called a kilobase pair. Okay, archaea, like bacteria, typically a single circular molecule, same rough size range, and then eukarya differ dramatically in that usually there is multiple chromosomes and they are linear rather than circular. Also, of course, there are many eukaryotic organisms that utilize sexual reproduction. So they actually have a diploid genome. So they have 2 copies of each chromosome. Haploid would mean they only have 1 copy. Now, most microorganisms, most eukaryotic microorganisms are generally haploid, but then many of them do have a sexual reproduction process, which is sort of optional. And that would include like the yeast Saccharomyces cerevisiae. Typically, eukaryotic genomes are significantly larger than bacterial or archaeal, ranging from 10 megabase pairs at the very low end to billions of base pairs. 1 billion base pairs is called a gigabase, okay? Including the human genome, which is roughly 3 billion base pairs, okay? So typically much larger. Typically the genes in eukaryotic genomes are much less densely packed than they are in bacterial or archaeal genomes, okay? So that's just sort of overview of genome organization. Now, I said that usually there's a single circular chromosome and 1 copy unless it's in the act of reproducing, in which case there's 2 copies. There are bacteria that have many more copies. D.radiodurans is an example of that. Each cell may have 4 to 10 copies of its genome, and that seems to be key to its radiation resistance, which probably actually has more to do with its resistance to desiccation than a natural selection for radiation resistance. There are other bacteria that have other quirks to their genome organization as well that we won't go through right now. By the way, you should be watching this hopefully before you're doing your bacterial genomes lab exercise for week 6, because this is sort of introducing you to the organization of bacterial genomes. There is another lecture for this on Wednesday though, okay? I'm sorry, on Thursday. DNA replication, something you should all be familiar with. I have links in here to some videos, and I encourage you to go through and watch these videos yourself because they will explain these concepts in a more visual manner. But basically, semi-conservative replication means that you pull the 2 strands of parental DNA apart, and each one of those strands then serves as a template for the synthesis of a new complementary strand, right? So now you have 1 old strand and 1 new strand. So each of the progeny from this round of replication has 1 old strand and 1 new strand. If you go through another round of replication, that old strand, the old strand from the original experiment will now have another new partner. And there will be 2 of the molecules that will just have completely new strands of DNA, right? So anyway, that's the point, is that each of the strands serves as a template for the synthesis of the new strand based on base pairing between the adenine and thymine and guanine and cytosine in each case, okay? How is that controlled specifically in bacteria? It turns out that replication of the bacterial chromosome only initiates at one site on the chromosome. So whether this chromosome is 500,000 base pairs or 5 million base pairs or even larger, there's only one place that replication is going to initiate. That's referred to as ORIC, for origin of chromosomal replication. Okay, so this is just some historical stuff. We're not going to go into how the details of what happened after, DNA was discovered and the structure of DNA was solved by Watson and Crick. Okay, so remember, there's this origin of replication. It's only a couple hundred bases typically, and there are 2 different conserved sequences in it, called here pretty mundanely the 13 base pair sequences and the 9 base pair sequences. The important thing here is that one of those, the 13 base pair repeat, serves as the binding site for a protein called DNA-A. So DNA-A is sort of the ultimate regulator of chromosome replication in bacteria. It binds directly to ORIC, and it is necessary for triggering the initial opening of the strands of DNA, the separation of those 2 strands. DNA-A is an ATPase, so it actually uses energy from ATP hydrolysis to help with that strand separation process. Okay, so DNA-A binds to that site in the origin. You have multiple molecules of DNA-A binding, and then you get a localized unwinding of the DNA, a separation of the strands. Okay, then the other key proteins for the initiation of replication come in. Okay, so there's a protein called DNA-C and a protein called DNA-B. The gene name for these would be lowercase DNA, uppercase b. The protein is uppercase D and A, and then uppercase B. At any rate, a helicase means that it pulls the strands apart. So once DNA-A has done its job, DNA-C and DNA-B can come in and bind at that origin, and DNA-B can then start expanding this region where the strands are separated. And those strands have to be separated so the DNA polymerase can actually get in there and start making the new strands of DNA-A. But it's critical to remember that DNA polymerase can't start that just from a single template strand. It needs a primer to be laid down. So it needs to have a few bases already there to add on to. Okay, so what makes that primer? The primer is an enzyme called primase. It's also called DNA-G because that's the name of the gene that encodes it. Okay, so once DNA-B starts pulling these strands apart, DNA-G gets in there and starts laying down an RNA molecule. DNA-G is an RNA polymerase. Okay, so it's really important that you get the functions of these proteins, the basic functions of these proteins right. Okay, now again, you're only going to have open book exams, so you don't need to memorize all this, but I do want you to focus on what each of these proteins is doing. Okay, so and then the other component that you have in there here is sort of a helper protein called single-stranded DNA binding protein that keeps this DNA unwound until the new strand has been laid down. Okay, so we separated our strands apart at the origin. We brought in the helicase to start moving along and pulling the strands apart, the primase to lay down the primer so that DNA polymerase can go to work. And then that's what you see here. So that last step was the initiation step for replication. This is what we call elongation. And this is where we actually get that key enzyme DNA polymerase coming in. Okay, so DNA polymerase, you don't actually see it here, unfortunately, because this is just focusing on the DNA. This is showing you what's happening at the so-called replication bubble, okay, where the strands are pulled apart, all right? And so it's important to understand that DNA replication is going to move in both directions from the origin. So this is going to be bidirectional replication. So you have a replication fork, which is the area where you have double-stranded DNA transitioning into single-stranded DNA because the strands have been pulled apart. You have a replication fork on both sides, okay? Now, DNA can only be synthesized by DNA polymerase in the 5' to 3' direction. RNA polymerase works the same way. It only works in the 5' to 3' direction. Okay, so if these were our original primers and they are more than 2 bases long, it's just being shown here for simplification. They're typically 10 to 20 bases long. Then the DNA polymerase acting off of this RNA primer is going to move in this direction. The DNA polymerase acting off of this primer is going to move in this direction. Okay. So that is what we call leading strand DNA synthesis. I hope this is something that you learned about in your earlier classes when you talked about both the structure and the replication of DNA, right? So this should be somewhat of a reminder to you. On the opposite strand, you have a problem because there's no polymerase that can just go like this or like this, right? The replication fork movement is being driven by the leading strand. And then what has to happen is the lagging strand has to be filled in behind it. And it can be filled in behind it because you can have primase adding new primers along the way, periodically, okay? So this region over here is called the lagging strand. And this is lagging strand synthesis. Likewise, over here is the lagging strand. The lagging strand is physically connected to the leading strand because they're just on different sides of the origin of replication, okay? So as the leading strand moves this way, essentially the lagging strand behind it is filled here. But more importantly, opposite of the leading strand, you have lagging strand synthesis filling in on the other side as well, okay? These little pieces of DNA are referred to as Okazaki fragments, okay? So the lagging strand synthesis happens in chunks, right? So each time a new primer is put down as this replication fork moves this way, then a separate molecule of DNA polymerase will be filling this in, okay? Now there is a process, let me go back for a second, sorry. There's a process by which these strands are, fragments are joined together. We'll get to that in just a second. Okay, now the last thing I want to mention on this slide is that we have a challenge in front of the replication fork, because as these two strands of DNA are being pulled apart, the region ahead of the replication fork is being wound tighter and tighter, right? This is all part of one big DNA molecule. Remember, this is a single circular chromosome. So all this is attached. You're just not seeing the rest of it. So you can't, this strand is, this side of the replication fork is not just free to rotate to undo the overwinding that replication fork separation is causing, right? So you actually have to have enzymes called topoisomerases ahead of this to relieve that overwinding. And that particular topoisomerase function is called the gyrase activity. Okay, gyrase activity is going to relieve that overwinding. Okay, and that's critical to have for progression of the replication fork, right? Okay, here's now where we connect this to antibiotics. In the main antibiotic that's used against bacteria that targets DNA replication is targeting this enzyme DNA gyrase, okay? It's also called topoisomerase 2. So gyrase is targeted by these molecules called fluoroquinolones, or quinolones is the original class. The contemporary class is called the fluoroquinolones because they're more stable and more potent. Okay, so fluoroquinolones include, for example, ciprofloxacin, norfloxacin, and levofloxacin. Okay, gyrase activity, as I just told you, is critical for DNA replication to continue. And so if you block gyrase activity, replication sort of grinds to a stop. It turns out that it doesn't just stop growth, they actually kill cells as well. That's a more complicated story. Not going to get into right now as to why the DNA actually gets broken up in the presence of fluoroquinolones, but it's a really interesting story that has to do with the mechanism of this topoisomerase DNA gyrase, right? Resistance to fluoroquinolones happens because there are specific amino acids in the gyrase primary sequence that form the binding site for the fluoroquinolones in proximity to the active site of DNA gyrase. And so you can get mutations in the fluoroquinolone binding site that reduce the efficiency of binding of the antibiotic and render these bacteria resistant. So this is a very different story than, for example, how beta-lactamases work. Beta-lactamases work by cutting up the beta-lactam antibiotic, but resistance to fluoroquinolones doesn't work by cutting up the fluoroquinolones. It works by changing the site on the target enzyme that the antibiotic binds to, so changing it by mutation, all right? All right, let's finish this up by talking about how the strands on the lagging strand are completed, right? So here you have the new strands. These are the Okazaki fragments. Basically, when the previous fragment gets to the next primer, right, the newly synthesized fragment gets to the next primer, the DNA polymerase 3, which is the main replication enzyme, falls off because it doesn't have the ability to remove that primer. There's a different DNA polymerase that comes in, POL1 comes in, and it has the ability to chew away this RNA primer. Then when it gets to the end of the RNA primer, it lets go, and another enzyme called DNA ligase now connects these new DNA strands so that it's all one continuous backbone. So this is sort of the wrapping up of lagging strand synthesis. So again, you have the POL3, which is a normal replicative enzyme, releases, lets POL1 get rid of the RNA primer and replace it with DNA. Then POL1 jumps off and DNA ligase connects everything, all right? So this is just sort of a table that is summarizing this in bacteria, what each one of them is done, and then sort of the equivalent functions in eukarya and archaea. I do not expect you to know these in eukarya and archaea. We're just focusing for now on bacterial DNA replication, okay? Final step in replication is that you have to separate the molecules apart because you've replicated two circular pieces of DNA. At the end, they're actually concatenated, they're attached to each other, and you have to resolve that. And so this happens at the so-called termination sites at the opposite side from the origin of replication. And there's a different enzyme, actually topo2 here, that's involved in that decatenation. Topo2 is DNA gyrase. There's a topo4 as well that can do this job, right? So that is the end of replication for bacteria. Okay, so I'm